think we can begin. It's 5:30, and so the International Center for Theoretical Sciences has instituted a lecture in the memory of Professor Abdul Salam. As you all know, he was one of the creators of the standard model of elementary particles. And for this work, he shared the Nobel Prize with Shelley Glashow and Steven Weinberg in 1979. But besides his extensive contributions to physics, he created the International Center for Theoretical Physics in Trieste, Italy, in 1964. ICTP is a unique institution with the mission of promoting science in the developing world. And it has contributed to the growth of science in India. Many of us actually have been benefactors of the existence of the ICTP. And it has also enabled this type of institution, the idea of this type of institution to propagate around the world. So the International Center for Theoretical Sciences of TIFR in Bangalore certainly in part was inspired by the presence of such a center. So for all these reasons, we have this Salam Memorial Lecture every year. The last lecture last year was given by Fernando Cuedo, who is the present director of ICTP. So this year we have invited Professor Ramakrishna Ramaswamy. Presently, he is the Vice Chancellor of the University of Hyderabad and also holds a professorship at the Jawaharlal Nehru University in Delhi. So he got his master's degree in IIT Kanpur. That's when I first got to know him. And I always wondered, how come a chemist knows so much mathematics? He used to know group theory and all types of things. Wonderful. Then he did his PhD from Princeton University and postdoc from Caltech. And his presently, as I said, at University of Hyderabad, his scientific work is very prolific actually. It's over 200 publications which span areas of nonlinear science, chemical physics, statistical physics, and computational biology. And in the area of nonlinear science, I think he's really one of the pioneers in India. He's a fellow of the World Academy of Sciences in Trieste, Indian Academy. He also is the Vice President of the Indian Academy of Sciences in Bangalore and the Vice President of the Indian National Science Academy in Delhi. He's the editor of publications of the journals of the Indian Academy, which have seen a real buoyancy in quality and distribution. And he's also the chairman of the National Council of Rural Institutes. He is very passionate about education, about really putting his mind to serving India in a very fundamental and definitive way. So I'm extremely happy that he accepted to come and deliver the Abdul Salam Memorial Lecture. Ram. What is this? So we have a memento from the ICTS for Ram. So thank you, Mansi. I've collected my goodies. I can go now. For that warm introduction, and also for setting the stage in a somewhat daunting manner. I'm very pleased to be here and to speak at the ICTS. And when Spenta asked me, my sort of stock reaction was, what could I possibly say that would be interesting to
ICTS people. And so when you made me think as to what I could talk about, and keeping some of the, keeping the occasion in mind, that I thought that I would talk a little about complexity and simplicity in biological systems. Spoiler alert should be given right at the beginning, that, the perspective will be that of someone from the physical sciences. So I would like to talk a little about what kinds of things one has been interested in. In a recent talk in Bangalore, Dan Stein, there's some way of seeing this over here. Okay. Dan Stein, the title of it, I think it was last week, I got an email from Shrikant. One of the deepest scientific questions we can ask is, how might complexity arise? Starting from simple, undirected processes subjected to physical and chemical laws, how can structures with complex shapes and patterns arise? This is a very deep question, and the talk itself, I have not had the privilege of hearing it, but as I discovered, he and I were contemporaneous at Princeton University, and he will be speaking in Hyderabad on Monday. But it's intriguing that people have been worrying about the idea of how complexity. In another context, Francis Crick, towards the end of his life, spent a lot of his energy worrying about or enunciating what he called the astonishing hypothesis. And the astonishment that he wanted us to appreciate is that at the end of the day, we are just a complicated assembly of molecules. The hypothesis itself is astonishing, maybe deep, may not, but it says that all living organisms, all their actions, responses, and even, and this is for him the important part, even emotions, are just the results of complex reactions of a vast number of fairly simple molecules. So in arguably the most complex thing that we have, the human brain and human behavior, attempts have been made to try to see it as to see a pattern of simplicity. And this is this is the theme that I would like to discuss. Many years ago in TIFR, Virasoro, then director of the ICTP, was talking, and he gave a definition of complexity, a simple definition of complexity. He said the more complex an object is, the more you can talk about it. I like that definition very much because I think it captures qualitatively what we mean by something being simple or complex. So there's not much you can say about that block of wood. Shoe, that is a shoe on the right foot. You can talk about the number of pieces that go into it. And this is one of the early designs of a bicycle. I thought it would be too simple to put in a motor car or a plane or something. So here's the way in which the pedals and the gears and the pulleys and everything work. And clearly this is a more complex object, less and less. And if we should want to describe it, just the description would take long times. This same idea was captured by Kolmogorov and Chaitin in their notion of complexity. And this is a definition of algorithmic complexity. The length, it's ideally described for strings of letters or symbols. What is the length of a program that is required, that you need to write in order to reproduce that particular string? So AB, 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 the program is write AB 4 times. Whereas AB, DF, G, K, X, Y, Z, etc. with no discernible pattern, the program will be write A, write B, write D, write F, and so on and so forth. So it's a computational complexity part, but again, it's the same thing. The more you can talk about it, the longer your program for writing it, the more complex an idea is, an object is. We're not very, very good about defining complexity in a quantitative manner for many of the objects that we have to be confronted with. We can talk about their dimensions, we can talk about this property, that property, etc. But a real quantitative theory of the complexity of life or of a living organism is not there. So what I'd like to do is to really talk about notions. One more notion of complexity that I will talk about is that of power laws, and then try to illustrate it with examples in the biological, physical sciences. About 20 years ago, or maybe a little more, there was a flurry of activity in an area which people called self-organized criticality. And this was a way for generating power laws in very simple systems by very simple rules. The archetypical example being that of a sand pile. Power laws are basically expressions of correlations that are very long in space and time, and arguably the more complex an object or the more complex a thing, whatever it might be, it behaves in a way which is highly correlated. There is some hidden design. But as people have discussed, power laws arise in simple ways as well, and I'll have some occasion to discuss that. Now, without getting into a quantitative definition of complexity, I will just appeal to this idea that the more complex the object is, the more you can talk about it, the more things it can do. By that very simple yardsticks, most biological systems are indeed very complex. Even the simplest ones are very complex. They do a lot of things, there are lots of functions, a lot of properties that we have to keep a track of. And in that sense, and I will not get more quantitative as regards complexity itself, given the multiplicity of functions they perform, biological systems are complex. And what I'd like to do in this talk is to talk about also features of simplicity, and in a variety of ways where simplicity arises within this complexity. I will try to illustrate it and touch upon both these aspects. So the first, okay, so I basically have three themes that I'm going to develop in this talk. And first I'd like to talk about the genome. As is sort of everybody knows by now, and the instructions, the blueprint, call it what you will, to construct any organism lies in the genome. And at least for the last 20 years or so, there's been a lot of activity in understanding the genome, although the structure of DNA itself was deciphered in 1953. And as everybody knows, it's a double helix consisting of 4 bases, A, T, G, and C, arranged in some pattern along a backbone. Now, the central dogma, the basic understanding of how life, or one part of how this all works, is that DNA is transcribed to RNA, and RNA is then translated into protein. So the DNA, a copy is made, and then the copy is interpreted. Now, as one can see, the DNA itself is a long linear sequence of A, T's, G's, and C's. And one might ask, one can ask a lot of questions about the DNA, but let me also point out that on Earth, as far as we know, and there's now enough experiments of a kind, life has a single origin. That origin was either the common ancestor cell, which was about 3 and little more billion years ago, and all of life has got sort of differentiated through evolution, starting from a single ancestor cell. Very recently, that's about 4 years ago, there was a test of whether in fact all of life is connected. Namely, there are 3 main branches of life on the planet: eukaryotes, us, archaea, which is extremophile bacteria, and bacteria, in numbers at least, the absolutely largest entities. And the question is whether they all form this kind of a tree, or did evolution occur in some other way. And at least now, in this particular paper that appeared in Nature 4 years ago, tests were done against various different alternates, and it seems that this is the most probable kind of tree that you could have, that there was initially a common cell, which first gave rise to prokaryotes, then to archaea, and then to eukaryotes. Anyway, be that as it may, the important thing over here is that the DNA sequence, the details of the sequence are important because the genes themselves code for proteins using this symbolic information. And the translation code is the genetic code. And this is a near universal code interpreted in groups of 3. Now this 3 there is a start, and there's a stop, and there's a way of reading this particular thing. I mean, this is all sort of extremely well known, but there is a point to it. The genetic code itself is very simple, and because all of life has a single origin, this is essentially a universal code. I mean, there are exceptions. The biological rule, which supersedes all biological rule, is that to every biological rule there's an exception. Okay, so you can just keep go circular in that argument till one is dizzy, but it's true. Almost all organisms use this universal genetic code. Now, within prokaryotes and eukaryotes, the genetic code. See, by now the genome sort of programs, they are able to sequence organisms with sort of essentially in a very repetitive. It's very simple. So it's a technician's job, and I know that it must be done hundreds of times a day. But if you take a simple prokaryote, the gene is a continuous bit of code. So starting at, over here, the annotation is given over here. This is from an entity called GenBank. And it says that you start at position 99 and you go somewhere more than 1,000. The gene is there. And so you look at position 99, and sure enough, you have the triplet which tells you that you start over here. And every successive triplet is a different amino acid. And so the code is just linearly written from position 99 onwards. And that's the protein that it makes. So the first complexity that comes about when you just look at the genome itself is that if you go to eukaryotic species, and we are all, any higher organism is eukaryotic, the gene itself is got by joining together all the parts that are in red. So there's a little part of the gene which is over here, then there's a gap, then there's more here, then there's a huge gap, then there's more here. And these things which are expressed are called exons. Those that are not are called introns, hearkening back to particle physics of some kind. But, okay, so nevertheless, you've got this. So the point of the complexity over here is you've got a code which is written in some script, but you've got little bits of it scattered all over a linear bit of DNA, with intervening portions which can be of arbitrary length. And as we will see, the complexity just begins with this. So in eukaryotes, the whole business of identifying a gene still remains a problem. Because of this essential complexity, the code is not linearly interpreted. It's interpreted with punctuation, except we don't quite know what the punctuation is. Okay. Now, the DNA itself has okay. So one of the questions that physicists have been looking at when interpreting genomic information is to ask, is there any correlation between a symbol over here and a symbol over here? Does it really matter? And sort of for nothing, we know that it has to matter, because DNA, after all, is it's a molecule that is formed, it's formed by evolution. So the correlations do matter, and what many people have been looking at is to look at long and short range correlations. I seem to have missed a, okay, I'm sorry, I missed a slide which I need to show, but I'll come to it later on. But the DNA is your double helix. That double helix gets wound around histones. Those histones are then stacked in a particular way. That entire stacking, all of that has got to be compressed inside every cell. So the DNA has to learn to bend. It has to learn to deform. It has to learn to twist. And these, the bending, the stiffness, etc., these depend on the precise nature of, is a string of this particular string, how bendable is that? That string of G's, how much do they bend, etc., etc. So the question of long and short range correlations is extremely important. The short range correlations are extremely important for another reason, that the chemical properties of DNA, what goes into producing a specific protein, that depends on the sequence of letters that are coming one after the other. So one of the ways in which people have tried to look at correlations is to ask, supposing I make a correlation function, what does this typical correlation function look like? Okay, so these correlation functions are, usual two-point correlation function that one would use in physics, namely, what is the correlation between the symbol alpha at position j and the symbol beta position k spaces away? Okay, the idea comes from linguistics, and you'll see that there's a lot of theme of linguistics that goes on in looking at sequences because of the formal similarity of language and DNA. But basically, this is looking at the correlations between positions. One of the things that we did in the early 90s was to take the same correlation function and convert it into a Fourier transform. Now that's quite easy to do because you just define the you define a Fourier transform in a fairly standard way. You basically convert your DNA sequence into a signal and compute this quantity. And you can do sort of interesting things with the Fourier transform. But if you take a gene, if you take any object which is known to be a gene and you take its Fourier transform through the formula that I just showed, what you find is that there's a very sharp peak at frequency 1 by 3. Okay? That frequency 1 by 3 is because the letters, the codon is 3 letters long. But essentially every gene in every organism shares this kind of property. Now, when I say every, I basically mean over 90%. I don't mean every. That's because of the, but you have it, it stands out as a very sharp signal, and this basically says that if you look at a string of DNA, given if you have an A over here at position 1, at position 4, with very high probability, you will have another A. If you have a T at position 2, with very high probability, you will have a T 3 positions away, and so on and so forth. The reason for this is evolutionary. We all go back to a single event of life that started 3.5 billion years ago. At least I believe that it's evolutionary. But this is what I would call an evidence of simplicity, because rather than having very long-range correlations, here you have a very sharp peak at frequency 1/3. If you take a stretch of DNA that is not known to be coding, that's what it looks like. It's absolute grass, right? Meaning the scales are the same, so that you can see that what looks like a very sharp peak on this scale, at the same scale for non-coding DNA, it is just complete grass. So non-coding DNA does not have this structure. Coding DNA has this structure. But DNA, of course, is not repetitive. So this is evidence of a slightly higher level of organization than that. And it will not go away. You shuffle the codons, you shuffle this, you shuffle that, you will always find. And so just to summarize, I suggest that the origin of this codon bias and triplet bias lies in the fact that somewhere in the very, very dim and distant past, we are descended from probably a very fairly simple, repetitive triplet polymer. It had some interesting structure in it, but that commonality of life, the commonality and the single point origin says that all of life really has that continuity, and that gives rise to this peak at 1/3. Now, this turns out to be a very good way to discover genes, because if you just find a portion of DNA that's of length 3n, and if it starts with the right symbol and stops with the right symbol, and if it satisfies this bound, it's probably a gene. And with that, I can identify something like 95% of all the genes that have been identified in bacteria till today. It's that organizationally simple. However, the coding story is not over yet. If you take a typical eukaryotic gene, which consists, as I already told you, of an exon, of several exons and several introns, it can be as complicated as this. The first level is that there can be many, many exons and many, many introns. The example I showed you was that of 3 exons and 2 introns. But there could be n exons and n-1 introns for you. What's even more interesting is that in different tissues of the body, different combinations of whatever is expressed will be made as a protein. So this is a gene called alpha-tropomyosin, and in certain kinds of muscles, you can either have this plus this plus this plus, you can see which ones are joined together. In smooth muscles, you have this part of it expressed. So the same gene in different portions of our body is making very different proteins. Where the instructions come from, how they get put together, the precise way in which this works is not known for all the examples. In 1 or 2 small examples, people understand how this happens, but the complexity is now a complexity of organization. And this eventually will show up in the way in which the complexity of organisms rises. So in the brain, for example, the same gene is expressed only in a certain part, a very truncated part of it. So these are all known examples of how alternate splicing works. So even though, and this was the big surprise for the Human Genome Project, we seem to have only 25,000 genes or so. But once you throw in all these variations, the actual number goes up to 3 to 4 times that number. So that is, again, an evidence of the complexity of genome organization. Okay, so since we have spent a lot of time looking at this 1/3 peak, we've seen, we've sort of played a lot with this particular idea. But the fact that the DNA, this is the slide I was looking for earlier. So the DNA itself is, on a scale of 20 angstrom, it's got its double helix over there. It gets wrapped around the histones. That gets wrapped around into these fibers. These fibers make these loops with some very nicely well-defined structural oscillations over here. And this whole thing gets tied up into a chromosome. Somewhere, the instruction that must land up at the end of the chromosome is tied into the primary structure. It's there inside the primary structure of the DNA. So the correlation between this eventual structure and this has to exist, because you have the wrong structure of DNA. Okay, may not be at every microscopic level, but there is a finesse over here. If you have DNA that cannot bend, then that bending will not even allow you to go around the histone. If you cannot have this thing bend, then it cannot wrap around, and so on. So there's a lot of structural constraints, and this gives rise to correlations between that are on DNA chromosomes which are very long, which can span several thousand bases. Right? One way again to look at it is to compute the Fourier transform of entire chromosomes. And this is for yeast chromosome. You do a Fourier transform, and there you come up with very nice 1 over F spectra. And for 100 years now, people have seen 1 over F spectra as evidence of complexity. Okay, this basically says there are long-range correlations. I mean, I've given you the biological reason why there must be long-range correlations. Here is just the mathematical evidence for these very long-range correlations. But the point is to be made that on the very long range, the system is complex because the first base has to remember the last, or vice versa. Okay? On the short range, you've got to have simplicity because you've come out of the old genetic code, and every third base has a high correlation. So there is some of that. Okay. There's a linguistic law called Zipfs Law, which basically talks about the usage of words. The word that is used most often in the English language, which is the, is used approximately twice as often as of, which is used again, is used 3 times as often as for, and so on. Okay? So if you take the rank of a word, that is the order in which the word is used in the English language, or in Tamil, or in Kannada, or in any language, and experiments have been done actually by my hero, Kosambi, in Kannada. The frequency of usage of a word of the nth rank inversely proportional to the rank. It works for DNA as well, except that you don't know what's the right kind of word. So you take triplets of letters, quadruplets of letters, imagining that they are words. It turns out that these word frequencies also follow a Zipfs law. But I'm more interested in another law, which has to do with the genomes. Our genome, the human genome, is 3 billion letters long. 3 billion bases in 23 chromosomes. Okay? Now, these three, different organisms have got genomes of different length. And here is the sort of the data that's available till today. If you take humans, that's where we are. Mammals up here, humans, if you just come down, will be 3 billion. It's always refreshing to know that newts and frogs are not very different. They've got DNA which is about the same length. Many plants, wheat and peas, matar, lilies, have got much longer genomes. Amoeba go to 10 to the 11, 10 to the 12, of that order. And so these are all the higher organisms. Over here in this corner are archaea, which extend from a little less than 500,000 to around 500,000 to a little less than about 6-7 million. All right? And bacteria, which go again from 10 to the 5 to about 10 to the 7. Okay? Now, see, this kind of a diagram hides a certain feature, but I just want to illustrate for you that when you look over here itself, the biggies, the big genomes, are organisms that are more complex, but not the most complex. Okay? This, in terms of complexity, in terms of what all we can do, nothing really beats the human. Okay. When you now look at the actual distribution of genomes inside bacteria and archaea, that's the actual distribution. It does extend from about 10 to the 5 till about 10 to the 7, but really speaking, there's not a lot out there at 10 to the 5, maybe 1 or 2 organisms, and very, very few at 10 to the 7, maybe again 1 or 2. And if you look at bacteria again in a little more detail, it turns out that for all bacteria, as you keep increasing the length of the bacterium, the number of genes slowly increases. Okay? So that's the red curve out there, the red line over here. It's almost a very nice straight line. The longer the DNA, the more the genes. Whereas if you come to the higher organisms, fungi and humans and so on, all these eukaryotes, the genomes themselves are 100 times longer, but the number of genes haven't increased that much. I mean, it's a more complex relationship over there. My interest, though, is it's in another law of linguistics over here. It's called Zipfs Law. Okay, Zipfs Law says the following. Okay, it was discovered by a German, and you'll see why that's important. The increase of a linguistic construct, for example, a sentence, it results in a decrease in the size of its constituents, the phrases. And this is an evidence, this is a linguistic example of correlations. Because if you go to make a very long sentence, I cannot really make a coherence. Let's assume that you want to make sense. Okay, so if you want to make sense and if you want to say a very long sentence, then one has to pretty much be sure that what I'm going to say at the very end, including the grammar, the punctuation, the full stops, etc., where all they have to come, and not be caught up in too much mean to say, we don't want all that stuff going on. So the correlation between the first word and the last word has to be very strong. And so too, it's easier to keep this organization if your phrases are short. So Zipfs Law says, if you have a very long sentence, phrases are going to be short. If you have very long words, the syllables are going to be short. Come and look at genes. If you have a very long gene with many, many exons, each exon is going to be short. If you have a very long genome with many chromosomes, each of the chromosomes is going to be short. Okay? This is just an evidence of the same kind of organizational principle, and it actually fits some kind of mathematical law of that kind. But our question, and this is something to which I don't have a very clear answer, actually because of some very recent work, not ours. Why is there such a sharp divide? Or why is there such a sharp divide on a diagram like this? Because really speaking, there's only 1 or 2 organisms out here. 10 to the 7 seems like you've got to stop. You've got to stop being a bacteria, and you see, because you look at this, it just comes hurtling down at 10 to the 7, almost no examples, and you go over into eukaryotes. Now, the difference between bacteria and eukaryotes is, of course, the fact that one has a unicellular organism and one is multicellular. But the question that one asks really is, why did this change happen? And this is almost one of the most profound questions in biology. And one suggestion that one is sort of advancing over here, which may well be wrong, but that if you just keep on increasing the length of a bacterial genome, the number of genes keeps on increasing, but there is a carrying capacity because all this reproductive machinery is based on proteins that decay. So just very simple balancing laws say that is it because of the physical size of the bacterium, or how did the eukaryotic cell come? And there are other sort of more bioinformatics-y kinds of questions one can ask. Okay. And in addition to the fact that life only originated once, it's also pretty clear that this transition from prokaryotes to eukaryotes only happened once. And there is really no convincing, there's no convincing theory which says why it happened only once, and because if it could have happened once, it can happen again. The origin of life, assuming that we were seeded by comets, Philae may have some interesting answers, Interstellar could tell you something about wormholes. I'm open on that. Okay, that perhaps it is such a rare event that it happened only once, that you just managed to land in a soup that was the one time. But this prokaryote to eukaryote transition, if it is just a question of size, it could have happened more than once, but there's no evidence that it did happen more than once. although very recently, there's been a paper as recent as less than a month ago, which basically said that one bacterium, or actually an archaea, ate up all, more and more of these other bacteria. So it sort of took them into itself, and what they call an inside-out origin of the eukaryotic cell. But nevertheless, this is an example of, to me, the whole origin of the complexity of life is tied into a question of this kind. Okay, right. These will the shorter ones. One of the things that I have found fascinating over the years is, how does one keep time? Within the cell, right? And within the cell, Mukund has worked a lot on this. The cell it's a warm, somewhat very crowded and very, very noisy kind of place. So whether it is your bacterial cell, that's a micron, or a eukaryotic cell, which is much larger, but really the inside of a cell is very, very dense and very, very crowded. In this, a whole bunch of reactions have got to happen fast enough for this astonishing hypothesis of Francis Crick to be essentially true, to be able to change one's moods fast enough, to do all sorts of reactions fast enough. How does the cell do it? Okay, and there are a whole lot of time scales in cells, in sort of living organisms, going from neural rhythms that are sub-seconds, cardiac rhythms which are a second, oscillations of calcium concentrations which affect so many processes inside cells, biochemical oscillations, etc., etc., circadian rhythms which are about 24 hours, and so on and so forth. Okay, and then there are, of course, much, much longer ones which have to do with years and so on. Okay. Now, many of these kinds of rhythms that happen in biology are a consequence of negative feedback, and circadian rhythms they're very familiar ones because people get jet lagged and whatnot when the circadian rhythm goes out of whack. This is something that we worked on. But what I want to draw attention to is that even though we have thousands of genes interacting in a very noisy, very complex place, somehow there is the imposition of a certain kind of order. And these kind of orders became apparent in experiments that have been done now for almost 15 years, where, ever since microarray experiments became available, people looked at the gene expression, that is the quantity of protein that was available, inside yeast at different points in time. And here the different points in time are marked along the cell cycle. These are just different instances, G1S, G2M. These are the 4 points in the cell cycle. So here is a gene expression that is essentially periodic in time. Is the same set of same data collected years later in another group. Again, you have this sort of oscillation. But what I want to draw your attention to is that each one of these images is made from a superposition of 25, 30, 50, 100 different reactions. Not all of them are the same reaction. That is to say, in this entire cacophony of different oscillations that happens in a cell, many of them start oscillating together. One form of synchrony that we find is when people walk together, they start walking in step. But here are entire reactions that are walking together, oscillating together in the same way. There are very large numbers of them, often similar reactions, but large numbers. And these kinds of, so the question is, how do very different processes get entrained in this manner? And this kind of behavior is what is called, in the language of dynamical systems, phase synchrony. Namely, oscillations of very different time scales and amplitude scales basically oscillate in the same way. And this is something that people have been studying over many, many years. But in some work that we did in 2007, we showed that completely stochastic processes, that is, systems that are dominated by noise, can nevertheless show this kind of phase synchrony. This kind of phase synchrony is actually something which is very well known in ecology. It's called the Moran effect. And the Moran effect basically says that if you have two populations of some species that presumably obeys very similar evolutionary laws, but these two populations are subject to different kinds of environmental fluctuations. So a population of sheep on the land and a population of sheep in an island, separated by some distance, then the correlation between the populations of the two species is really equal to the correlation between the environmental fluctuations. Okay, so here you have got two different populations and two different kinds of environmental fluctuations, but the population evolves with the same noise, with the same law. So the change in these populations is just equal to the correlation between the two noises. In the language of dynamical systems, this is what we call generalized synchronization. Now if you have a common noise driving two different processes, it turns out that these two processes start getting correlated. Okay? And this is at least one of the things that we believe is responsible for the extreme coherence that you find inside a cell between diverse processes in the presence of noise. The point is that the noise is common to all the processes at the same time. And depending on how these various interactions can arrange themselves, it turns out that very different patterns of time and timekeeping can be devised. And to some extent, this is how we think that cells are able to, to understand when you have an oscillation of a day's length, less than a day, and so on, is just by exploiting different patterns and interactions like this. Okay. In the last few minutes, I'd like to tell you a little about how networks and systems are also examples of complexity and simplicity in biology, using the paradigm of systems biology. And I can see one of the Hyderabad students from systems biology here. So the idea is that you integrate biological experiments with quantitative analysis, computer science, etc., to try to give a framework for how different biological processes are interacting. Okay, now this is a vast subject, but I'd like to basically focus on one example. One of the themes that I've been emphasizing is that we are just a bunch of molecules. We are a bunch of molecules which interact in interesting ways and have a whole set of important reactions. And these reactions are mediated by other molecules, so genes, etc., etc. Now, all this comes together when you try to understand the behavior of a system. And today, the paradigm that is employed is that of networks. If you take Drosophila, the total number of genes is only, it's about 6,000 or 7,000. And in the humans, it is about 25,000. So there's not much of a difference, really speaking. It's not a factor of 10. It's a factor of 3 or 4 that we differ in terms of the number of genes. What is very different about the genes in our systems as compared to the fly is that each one of our genes, okay, what's drawn over here is an interaction pattern between the genes themselves and the proteins, how they interact. If they interact in a significant manner, you draw a link. But the genes of the fly and the genes of humans have a very different pattern. Our network, the intrinsic network of genes in the human, is much, much richer than it is in simpler organisms. So the complexity of human behavior and the complexity of human response may not be in the fact that we have many more genes. As we've seen right at the beginning, we have smaller DNA. It's smaller DNA by a factor of 10 or 100 than organisms like ferns and amoeba. But the richness of this network is what gives rise to the rather complex behavior that we have. So in the last few minutes, I'd like to talk about the systems biology of a disease like cancer. Now, a disease like cancer is properly considered systemic rather than a single, something that is due to a mutation or by some unique biochemical event, because there's a very complex and very individual response that seems to depend on the environment of the organism. In this case, we're mostly human cancers, but also in mice or plants or what have you. So it depends on the environment, it depends on the genetic composition of the individual. Two identical twins do not have the same, they may have the same predisposition to cancer, but they may not have the same outcomes when it comes to actually contracting the disease. So this is a disease which people have studied for a long time, and some parts of the disease network are really extremely well studied by looking at microarray experiments that people have been able to do for the last 15 years or so. So you go to a diseased individual, take a sample of tissue, and find out which genes are more expressed or less expressed than the genes of a normal individual. Of course, it's thrown up whole ideas of what is normal and so on and so forth, but nevertheless, the comparison is between normal versus abnormal, and you're looking now at the molecular level, you're looking at the response of literally thousands of genes. From that, you try to construct what are all the interactions that are happening. Now, individual experiments, molecular biology has taught us that there are a whole set of proteins that are important. The central player is something called P53. P53 activates another gene called MDM2, which represses P53. So the two of them, one promotes, one keeps it down. You maintain a balance. In each one of us, in every cell of ours, that's happening. When that goes out of whack, bad stuff tends to happen. In particular, if for whatever reason you go out into the sun and you get DNA on your skin that breaks, that sets off a signal which is sensed by P53, and it chooses to do one of three things. It will either say that the damage is too great, let's stop the whole thing, and let's not choose to reproduce this particular cell. It's called apoptosis. Or it says, let's stop the cell cycle, repair, and then get back to what you were doing before. Or if things have gone too far, let's stop cancer actually forming. And that's the third pathway. It's called inhibition of angiogenesis. Okay, so these are decisions that are made within the cell, molecular level decisions that have to be made in real time. And how does this actually happen? It's part of the fun of systems biology. What we've been working on is the fact that there are multiple networks. There's a network of genes, there's a network of proteins, and you can control a gene by controlling the protein and vice versa. The real life analogy is that if you want to communicate, you can communicate by the phone or by email, but if the email server is down, you've always got the phone, and if your phone network is not working, you may be able to get through on the email. So, multiple networks are really the connected way in which not only the social world lives, but also the genetic world. And these are typical experiments where these are all the genes. Each one of these little pixels is the expression level of a gene, differentially expressed between normal and diseased individuals. And these are about 500 odd genes and 120 individuals. And these were among the earliest works, bioinformatics works, that showed that breast cancer was not a unique disease, but was actually 5 or more diseases, all that just shared the location of the breast and were therefore called breast cancer, but they were actually distinctly molecularly different diseases. So that's what you learn with analysis of this kind. But you can do a little more. You can construct these networks. You identify the disease genes by looking at the genes that are implicated in this disease, use microarray information, and construct the network. These bonds show the interactions. This is a schematic, but you get the idea. So this shows the interactions between different groups of genes. And within this, they turn out to be what are called modules, groups of nodes that are linked a little more closely with each other than they are with the remainder. So here is one module, another module, and so on. And the interesting thing about a module is that you can actually try to take out the entire module by destroying important components inside this. This gives at least people who look at this kind of data ways of trying to identify targets. But this is at the gene level. Each of these genes actually expresses a protein. So you can look at the expressed proteins and ask, what are the other proteins that they interact with? So when you look at the protein level, you can find out all the interactions between various proteins. And now you have the real picture of the network inside the cell, where you've got all the genes that are interacting, all the proteins they are producing, all the proteins they are interacting with, and potentially affecting any one of these, if you choose it intelligently enough, is enough to disrupt the network. And when you disrupt the network, you potentially have a way of addressing the disease, either understanding it or curing it or whatever. Okay. So you can look at the protein space and so on. Now, this kind of, these are not the only two networks. There's the other networks. There's a network of metabolites. There's a network of lots of things, microRNAs, all sorts of stuff is there. So these networks, understanding not just the network, but the interaction of various networks, what people sometimes call the network of networks. That's what we describe in these two papers. And like I say over here, affecting one of these modules is like affecting the entire group. And this could give interesting ways of addressing diseases like this. Okay. So let me summarize. The three themes that I've tried to look at is, to give examples. First, in the simplicity and the complexity in the organization of genomes, the short range, the long range correlations, and the sharp size boundary, the unity of life, etc. These are all different aspects of simplicity, complexity. When it comes to the dynamics, there is coherence and synchrony in a very stochastic and very noisy environment. The fact that there is a commonality to all the noise, I believe, throws up the commonality brings out these very strong correlations. When you look at networks in the cell, there's complexity and interaction between these several networks that interact within the cell, and this gives rise to complex responses. And clearly, you can affect things at one level by affecting the networks at various levels. You can kill off the metabolic supply of glucose, and then there's no protein production or anything going on. So there are ways in which each network interacts with the others. Basic fact is that nature, over the last 3.5 billion years, has basically been tinkering. It started with something that worked and kept playing with this pattern over and over again. When it comes to the genome, it is a fact, and a lot of work goes on in biology and in bioinformatics. Genes are conserved across entire species. The same type of gene does the same type of function everywhere, and this is over billions, several millions, if not billions, years of evolution. So that's one aspect. That's one of the reasons, I believe. The same thing holds true for the networks, the network interactions, the motifs, the modules. If it works, nature tends to reuse it. And so the time pattern, you see, we have got a little component. The systems or the synthetic biology approach to life is you've got a module, you can use it. And nature is the best user of the same module over and over again. The actual networks themselves tend to be very modular. They are not highly connected, but they're deeply connected, and those are then loosely connected. So nature tends to use this kind of architecture to ensure a certain type of robustness. I've just touched upon various aspects of this. And what I'd like to do is to summarize by going back to 1996. Hans Frauenfelder was asking, what will the physics of the future look like? What will the student in 2096 learn in his physics course? So he'll learn quantum mechanics, classical mechanics, thermo, etc. But which field will be active in research? And says that my guess is the physics of complex systems will occupy center stage, and that biological physics will dominate. And a few years later, Laughlin and Pines, the central task of theoretical physics in our time is no longer to write down the ultimate equations, but to catalog and understand emergent behavior in its various guises, including potentially life itself. Physics of the next century is the study of complex adaptive matter. And we are witnessing this particular transition. To my JNU antecedents, one must give a salam to Abdus Salam, because he systematized so much of theoretical physics in such a deep and fundamental way. It is not clear whether there is a standard model of anything in biology, but out of the complexity of particle physics that existed in the 60s, it took a special mind to see that there was simplicity in it all. And that, I think, is something that he communicated. I chose the day for my talk because Penta asked me when I'd like to come and talk. I looked up Wikipedia and found that today is his death anniversary. So it's particularly appropriate that I'm able to give that talk here today. I'd like to thank you for this opportunity and leave with a quote from Niels Bohr, also to acknowledge one's physics antecedents, that the opposite of a profound truth, that there is complexity in biology, may be another profound truth, that there is also simplicity. Thank you. Sure. Yes, Abhishek.
So in this, sequence correlations, other than this one-third structure, are there other interesting features?
We don't know how gene regulation happens over very, very long distances. So we don't know what things to take the correlation functions of. The most obvious ones are the one-point correlation functions. And even if one was to take 2-point or higher point correlation functions, it's not entirely clear what those objects should be. So it's an open and interesting question, but we are not clear what those, like I said. People have looked at the correlation between 3-letter words, 5-letter words, etc., etc. Gene Stanley, for example, had an industry which did that.
So, just a follow-up question.
Yeah.
You said at some point of the talk that, so I guess so the best explanation for this 3-letter repeat is that this was sort of something that was evolutionarily picked up and then it's been propagated. Yeah. But are there ways one could think of validating that particular possibility, or that?
See, people are worried about what the ancient DNA or RNA code was, and the best kinds of explanation really go to some polymerization reaction that happened in some clay on the shores of some sea somewhere. the early catalysis of the RNA world hypothesis just says that there must have been some polymeric molecule. The simplest polymers are repeats, and therefore, yeah.
Just to follow up, I mean, 99.99% of the human genome won't have that repeat. It's only in the coding regions.
It's only in the coding
so
region.
It's just a fact about why isn't it just a fact about triplet codons and repeated amino acids in proteins?
Because true. The point is that it is not an exact repeat. It's not even an approximate repeat.
But we sort of know the origin. It must be just because of these set of letters for this particular protein, and if you have the same, I mean, there's some detailed reason. It is codon triplets plus a detailed.
No, it is definitely the codon triplet, and the fact that if you in the non-coding region it could tolerate a whole lot of mutations, but in the coding regions it could not. So there's, the fact of the code puts a very strong constraint on what are the kinds of substitutions you can have.
But nothing to do with billions of years of evolution. It's the structure of proteins.
No, it is the structure of the codon. The fact that you have if you have a 4-letter codon, you'll have a peak at 1/4, or you'll have a 4-base correlation. True. But it also is indicative of that there was no independent origin of life with some other sort of,
How do we know that? I mean, life may have arisen several times independently and then just died out in all.
That, of course, I mean, look.
But is there any fossil evidence or anything to suggest?
These are molecules with half-lives of few seconds, so it wouldn't survive in the wild.
Well, if there are no questions, let's thank Ram again
Thank
for a
you.
beautiful talk.
Thank you.